After finishing her PhD, Cristina moved to the United States to David Scadden's laboratory at Harvard Medical School for her postdoctoral fellowship. There, Charles Lin introduced Cristina to an exciting new technology at that time, which is called intravital microscopy, or IVM. I believe that David and Charles are joining us for this lecture. If you have been able to join, welcome to you both. IVM allows the imaging of cells of a live animal at high resolution. As a postdoc, Cristina pioneered the application of IVM to stem cells in the bone marrow. She was able to track these blood-forming cells in living animals over time. Her discoveries were reported in a landmark publication in Nature, which now has over 850 citations. In 2009, Cristina moved back to London to take up a lectureship in the Department of Life Sciences at Imperial College. She rose through the ranks at meteoric speed, becoming a full professor within 10 years. As you will learn from Cristina's lecture, she has not only used IVM to transform leukemia research, she has also opened up new avenues for treatment. Cristina's research has been recognized by the receipt of several prestigious awards, including the Academy of Medical Sciences Fulks Foundation Medal, which is given every 2 years to a, quote, rising star within biomedical research, and also the Carl Zeiss Lecture of the German Society for Cell Biology. Previous recipients of that lectureship include 2 Nobel Prize laureates. So no pressure, Cristina. But I shouldn't only focus on Cristina's research. She is also an inspiring and effective teacher. As an example, she convenes a very popular final year undergraduate course on stem cells, regeneration, and aging, in which she engages the students in discussions about the ethical implications of stem cell research and therapeutics. Cristina is a passionate ambassador for her profession and is very active in outreach activities. As a volunteer for the Inspiring the Future scheme, she helps to encourage young people to aim high. I am delighted that there are school pupils amongst the attendees at today's lecture. Cristina, your outstanding achievements were deservedly recognized by promotion to Professor of Stem Cell Biology in 2019. I am delighted to invite you to deliver your inaugural lecture entitled, on the screen, Battles in the Bone Marrow, Leukemia and Blood Stem Cells Under the Microscope. So over to you, Cristina.
Thank you, Anne. It is really a pleasure to be presenting here, even though online, but I know that many people are following, and your introduction has been really kind. Many thanks for your words. What I will talk about today is what has become my absolute favorite cell, and this is the hematopoietic stem cell. Now, we have a few thousand hematopoietic stem cells, and these few rare cells are able to sustain production of red blood cells, white blood cells, and platelets throughout our lifetime, day after day. As David Scadden put it very nicely a few years ago, hematopoietic stem cells produce more cells than there are stars in the whole of the universe put together. So they really do a quite astonishing work. Not only they do this, but they do it based on demand, so that if, for example, we are having an infection or there is some other kind of stress occurring to the system, the stem cells can adapt and their output changes so that we can have more red blood cells if needed or more white blood cells if needed, etc., etc. Now, hematopoietic stem cells were also the first one used in the clinic through bone marrow transplantation, even before we knew that there were hematopoietic stem cells. But one thing that is very interesting about them is that they only work when they are inside us or inside their host. We are not able yet to culture hematopoietic stem cells in vitro, and expand them and make them generate more stem cells and the appropriate amount of blood. So what is very important is that we understand where hematopoietic stem cells are within the body. And we know that they are inside the bone marrow, where they interact with very many cell types. So today, I will be taking you in a journey inside our bones, looking at the bone marrow, where we have a very complex bone structures that host not only blood stem cells and their entire progeny, but also a number of different cell types. We have endothelial cells, and we have multiple types of stroma cells that all have been shown one by one to cooperate together to regulate hematopoietic stem cell function. And in particular, in my group, we are looking at what happens with the interactions between hematopoietic stem cells and the bone marrow microenvironment during the regeneration, for example, when hematopoietic stem cells are transplanted. And in this case, few stem cells are able to regenerate the entire hematopoietic system of the recipients. We also study situations of infection, and we study leukemia, which is the main competitor of healthy hematopoiesis. And if this was a normal talk, this is where I would now start talking about some of the more recent projects that have been developing in the lab. But because this is the inaugural lecture, I think it's actually more important that perhaps I take a couple of step backs and I first tell you about how I got here. And then we will talk about this specific research a little bit more in detail. So I grew up in Torino, in Italy, which is actually a very wonderful place. And it is located just in between the Alps on one side. And so, as you can see from the image here, and believe it or not, from half of the streets in Torino, you can see the Alps at the end of the road, providing really beautiful view. But it is also quite close to the sea. And so I spent my early years with summers spent either on the Alps or by the sea, where, again, I could have nice view of the environment all around. And I kind of like to think that this being used growing up, looking at open spaces and wide environments, sort of stayed with me and left me with the interest of looking at the complexity and the variety of cells that interact all together within the bone marrow. But anyway, without getting too cheesy, I need to mention a few people that mentored me when I was in Torino. A number of professors I worked with, because I graduated in medical biotechnology, which was a very new course that had just been started by Professor Lorenzo Silengo, who was recruiting us as first recruits to the course with a sales pitch that through this course we would be able to discover and generate the new therapeutic approaches that eventually would be used by doctors. And instead, if we were to graduate through medicine and become doctors, we would essentially apply the results of the research. Because I had become interested in research growing up, I thought this would be a good course for me. It was a very unusual course in that we spent a large amount of time, much greater than any other undergraduate courses existing in Italy at the time. We spent a huge amount of time in the lab. And so a laboratory that was run by Professor Silengo, Professor Altruda, and Professor Tarone was the first place where I did mini preps, and I cloned bacteria and used pipettes, and it was really a wonderful place. And then through Professor Maria Flavia Di Renzo, who is also logged in today, so hi, and thank you for being here. I was actually put in touch with Alberto Bardelli and Enzo Medico at the Istituto Candiolo, which was the biggest cancer institute in Torino, and that is where I eventually developed my thesis. Now, during my undergraduate years, one thing was becoming very evident, and it was that medicine was changing, and a new type of medicine was developing, and this was what we now call regenerative medicine. And this was all driven by discoveries in the stem cell field. And so I decided that stem cells perhaps were the way to go, and they were the cells that I would want to study. So again, with mentoring from Professor Di Renzo, I had a look at the literature. I looked for types of research that I found exciting and interesting, and I sent out a couple of emails. And Fiona Watt in London was brave enough to reply to me and invite me up for an interview and hire me actually on the spot, which left me very surprised, actually, at the time. It took me more time to decide to do my PhD in London than it took Fiona to offer me the position. And that really started the beginning of a journey that was absolutely fascinating. We were based in the Cancer Research UK London Research Institute in Lincoln's Inn Fields in London. And during those years, I met a number of wonderful people, a number of lifelong friends. We had wonderful lab retreats, which I'm still trying to reproduce now with my group whenever we can, obviously not this year. We had a number of nice habits. For example, at Christmas time, we used to go ice skating at Somerset House. I think this picture is from 2002. And actually, I was very proud when in 2017, my group suggested that we went to Somerset House to go ice skating. And so here we are a few years later. In terms of research, what we were doing was to engineer a way to control the activation of a gene called beta-catenin in the epidermis of mice, so that we could activate hair growth, and we could also generate, initiate the generation of entire new follicles. This was very exciting, but what was truly fascinating for me was the approach that we had to the problem. We used to look at the skin of these mice, and we used to use quite a lot of microscopy, and especially confocal microscopy of whole mount preparations of skin, as you see in the image here on the right. Now, these images were absolutely beautiful and fascinating, but they were somehow also frustrating for me, because in these images we could see interfollicular epidermis here in the background. We could see hair follicles. There are 4 here in this image. They had sebaceous glands halfway through, and the cells highlighted in green here were cells that we knew were particularly quiescent, and we knew that somewhere in this region where we had the green cells, there were stem cells, but we couldn't quite point at the stem cells. So we could activate them, we could do things with them. Epidermal stem cells could be grown in culture and be expanded, and yet when we looked, when we wanted to isolate them and know exactly which cell would be the stem cell, that was very hard to do. So by the end of the PhD, I was thinking, I absolutely love microscopy, but I really want to know which cell is the stem cell. And so I became interested in the hematopoietic stem cell field, because there we were able to use, people were able to use cell surface markers to identify very rare hematopoietic stem cells. I went on a number of interviews for my postdoc, and that is where I met David Scadden in Boston. And I remember at my interview, after showing the microscopy images that I had generated with Fiona, he told me pretty much this. He told me, I think with you, we can actually find where hematopoietic stem cells are located, and I know who can help us find them. This was a big question at the time, because, as I said, we were able to use cell surface markers to identify hematopoietic stem cells, but this was all done after harvesting bone marrow tissue, and so it was all done in a tube. So we knew where the hematopoietic stem cells were located in fax plots, but actually we had 0 ideas where within the bone marrow they would be, which I actually thought was quite surprising for a field that was as developed as that of hematopoietic stem cells. So David paired me up with Charles Lin, and so a number of years started between being at MGH, Harvard Medical School, the Harvard Stem Cell Institute, and Charles was based at the Wellman Center for Photomedicine, which very conveniently was only a couple of floors upstairs from where David's lab was. We had again a number of years of very nice activities. We had again lab outings. I met another number of lifelong friends. Here you can see David taking us on a yacht sailing across the Boston Harbor. And one other very interesting thing that I could do was to actually start working with Charles' prototype microscope that did not look like any microscope at all, because I wasn't used yet to seeing prototype microscopes. I was only used to the commercial ones. But yet, by using this very complex machine, which one day did blow up on me, actually, I will forever remember that. But the amazing thing of that was that because Charles was building it, but Charles and his group were able to build it, they were also able to fix it. And so within a couple of hours, actually, we were able to do our microscopy. Now, at the time, we had to work with a model that was based on harvesting hematopoietic stem cells from donor mice. We had to label them with a chemical ex vivo, and this would be a highly fluorescent chemical, and then we would inject them into recipient mice. And we were able to look for them in the bone marrow that is contained in the top of the skull of the mice. So once we put all our efforts together, we were able to start intravital microscopy of the hematopoietic stem cell niche, which is where I have remained since those amazing years. And once our efforts came to a result, this was one of the images that we could take. In white here, you can see a hematopoietic stem cells sitting inside the bone marrow of mouse. We know that we are in the bone marrow because we are in an area that is surrounded by bone, which is in blue here. And we can see that this cell is localized close to these green cells, which are bone-making cells or osteoblasts. And in red, we can see a vascular dye that is at the moment filling the bone marrow because we are looking at an irradiated recipient. And the interesting thing about this image is that this stem cell is here, and we captured it only a few hours following its transplantation. So we could see with our eyes how efficient and how quick hematopoietic stem cells are at reaching the bone marrow cavity. And following this, we could do a number of measurements. And so, for example, we saw the functional hematopoietic stem cells tend to localize sometimes directly adjacent, like in this case, and sometimes just nearby, but always very near osteoblastic cells, these cells in green. We could see that their progeny was more widely distributed across the bone marrow space, and that also malignant cells would be much more widely distributed. And essentially, I was hooked. And I did continue to use intravital microscopy in the following years once I moved to Imperial College and my group started growing there. So I'll switch now to work that we did at Imperial College. One of the first things we developed was to add a couple of tweaks to the way we were imaging the cells in the bone marrow. And so we became very good at keeping the mice very stable within the microscopes while they were anesthetized, so that we could acquire this type of images where we could see the entirety of the bone marrow space contained within the top of the skull or calvarium of the mouse. And just to give you an indication, a little bit of orientation here, the nose of the mouse would be here on the left, the neck would be here on the right. We are seeing in blue vasculature, and there is one large vessel, which is called central sinus, that runs from the front to the back in this space. And on the two sides, you can see much smaller vessels that are organized in this honeycomb structure. And wherever we see these small vessels, these are the sinusoids that crisscross the bone marrow space. So this is where we are looking at the bone marrow space. The other cell that we see here in green are our osteoblasts. And now we can start appreciating how these cells are organized in patches that are fairly evenly organized across the space. Another thing that was very important for us was to become, on one side, quicker at acquiring the images, and on the other side, also quicker at analyzing them. And so we developed, or we adapted a number of computational approaches that allowed us to semi-automate the image analysis. And for example, we became much better able to measure the position of stem cells and other hematopoietic cells of interest relative to other stroma components. Now, an interesting thing of these images is that even though we are seeing a large area of bone marrow here, these images still have very high quality, and so we can still zoom in and have single cell resolution information about specific areas that we may be interested in. Not only this, but we can then focus on specific areas that we're interested in, and we can time-lapse them for a number of hours. And this is the kind of work that we continue to do, really following the initial steps of the experiments that I was performing in Boston. And this is work that has been incredibly challenging because we need to harvest cells that we can identify with more and more markers, but this makes them more and more rare. So we have fewer and fewer cells that we need to be better and better at labeling, and then we need to follow. So this work has been driven over the years, especially by Rima and then Sarah in the lab. And we've been having some help with that analysis from Chufan Lee in bioengineering and Ben Partridge in his group. What you're seeing here is, in a way, the follow-up of that large image that I showed you earlier of one of the first HSCs that we saw in the bone marrow. We are now looking at hematopoietic stem cells 2 days after transplantation on the left and 4 days after transplantation on the right. And we could start appreciating how these cells have complex interactions with the microenvironment. They may not move very long distances, but yet they do move a little bit. Some cells, like this one on the left, more than others, compared to, for example, the one on the right. But even the ones that don't move, they still have small protrusions and they have dynamic interactions with what surrounds them. A large amount of proliferation takes place in the first days following transplantation, and so we have more cells by day 4. And we also know that these cells are starting to differentiate. So what this type of work is offering us is really a window that allows us to understand the biology that is happening at the beginning of the regeneration of the hematopoietic system. We also know that by day 8 following transplantation, we now have very large patches of cells within the bone marrow cavity. And also within these patches, the cells are still very active. And we can see them moving around. In this movie, in the top part of the movie, you can see cells moving very fast. These are in circulation. There is a blood vessel here. But you can see that the other cells, which are in the parenchyma, are also wriggling around and being quite dynamic. And so, in fact, with Chufan's help, we are starting to apply active matter physics to understand the dynamics that are taking place within the bone marrow and that lead to regeneration. Now, one of the things I became very interested in since moving to Imperial College, and because within South Kensington campus, there were a number of groups studying a number of pathogens. One of the things I became interested in was understanding what is the impact of infection on hematopoietic stem cells. We knew that infection does affect not only immune cells, but also progenitor cells. And around 2010, 2011, so soon after I moved to Imperial, the field started gaining an understanding that even the stem cells themselves were able to directly sense infection and respond to it. So we first teamed up with Nikki Gunaris and Murray Selkirk, who were experts in a little nematode called Trichinella spiralis, and this is the agent that causes trichinellosis. And with help from the imaging facility from Martin Spitaler and from mathematician Ingo Röder in Dresden, 2 people in the group, Narges Rashidi and Mark Scott, were able to capture the behavior of hematopoietic stem cells harvested from mice that were either healthy or infected with Trichinella spirali. What you see on the left is now in red an HSC that was harvested from a healthy mouse. It was injected into a recipient mouse, and we're looking at it 1 day after injection. And you can see that the cell is there. It's making this little wriggly movement, but not so much is happening. On the right, instead, when we look at the stem cell that was harvested from a mouse that had infection, we can see that now the interaction with the bone marrow microenvironment is completely different. And the HSC is now motile, and effectively it is interacting with a larger niche. We also happen to know that these stem cells from the Trichinella infected mice have an improved function, and they are better at reconstituting their recipients. And in fact, still to these days, we are investigating what are the mechanisms that make stem cells from Trichinella infected mice better at reconstituting recipients. But what we importantly learned from this study was that really infection causes something deep at the level of HSC niche interactions, and these change. Now, there are some complications with studying mice infected with Trichinella spiralis by microscopy, which is that we were not able to put to directly analyze the infected animals with the microscope. And so, looking for a different type of pathogen, we stumbled onto a different disease, which we have been studying now for a number of years. And this was in collaboration with Bob Sinden and Andrew Blackborough when they were both at Imperial College. And a visiting student, Letizia, was with us for about a year. But with her, we were able to implement what Bob and Andrew were already doing, which was to work with mice and mosquitoes and replicate the entire life cycle and the entire pathophysiology of malaria in mice by using mosquitoes that carry the parasites and would bite mice and lead to the development of malaria. The parasite that was used was the mouse. Was a mouse parasite that leads to malaria, which is Plasmodium berghei. And what Letizia was able to see was that indeed, in this model of natural infection, hematopoietic stem cells were sensing the infection and they were becoming highly proliferative. Now, after Letizia left, this project was picked up by Miriam, who recently completed her PhD. Well, when I say recently, it's actually already a couple of years ago, so time really flies. And in her investigation, she had wonderful collaborations from Ken Duffy, who's a mathematician based on Maynooth University, Bertie Godgens in Cambridge University, and more recently also Tiago Luis, who joined the department a couple of years ago. What Miriam investigated was not only the simple notion that stem and progenitor cells would proliferate during this severe infection, but she actually looked at what was the turnover of specific population of stem and progenitor cells. And so with these mathematical models, she could highlight that when we looked at early progenitor cells, we could in fact see with the maroon line here that the turnover of the population was highly increased. However, when we focused on a population that we knew was very highly enriched for very primitive hematopoietic stem cells, yes, the turnover of the population was increased, but not to such an extent that would make us think that the stem cells were proliferating too much and therefore becoming exhausted. So this prompted us to think that perhaps other mechanisms beyond simple proliferation were affecting hematopoietic stem cells in this system. And when we perform single-cell RNA-seq analysis of stem and progenitor cells from healthy and infected mice, we could see that normally we could find a significant proportion of cells that would score highly for what we call the HSC score, which is a signature based on the expression of thousands of genes that is linked to stemness for hematopoietic stem cells. In these graphs in particular in here, you can see that each dot represents a cell, and so the red cells are a significant amount within this wider population of stem and progenitor cells. When we looked at the same population within the infected mice, however, the red cells had become very rare. And what this told us is that something very deep was happening at the transcriptional level in the hematopoietic stem cells, and we were perhaps either losing them entirely or they would not be able to perform as stem cells anymore. And in fact, Miriam could prove that if we were to transplant hematopoietic stem cells from the mice that were infected with Plasmodium berghei, these cells would not be able to reconstitute hematopoiesis. She also looked at the mice that carried Plasmodium, and she could see that while in healthy mice, with our usual tile scan images, we could see the whole area of bone marrow, and we could see many osteoblasts in greens, which I'm sure you recognize now. By day 7 of infection, the osteoblasts had actually gone, and this was happening through systemic thinning of the osteoblast population, so we would get fewer and fewer osteoblasts over time. And by day 7 of infection, when we had lost the osteoblast, we had also lost functional hematopoietic stem cells. So from looking at these images, we started wondering whether we could do something that would affect the bone marrow microenvironment and the osteoblast, and whether this could potentially rescue hematopoietic stem cell function. We knew from a number of years, in fact, from pioneering work from David Scadden's group, that osteoblasts were linked to functional hematopoietic stem cells, even though indirectly. And somehow in our studies, we kept seeing functional hematopoietic stem cells near osteoblasts. And so we thought, let's try something and intervene. And to cut a long story very short, what we learned was that if we pre-treated mice with parathyroid hormone, which is a bone anabolic hormone that activates osteoblasts, and for the last period of the treatment and throughout the infection time, we also treated them with a quencher of reactive oxygen species, then we could actually partially rescue the function of hematopoietic stem cells. So in these graphs here, you see the ability of stem cells to engraft irradiated recipients. The black line shows the result from healthy mice, and the engraftment is good. The maroon line shows the very poor engraftment that we see from cells from infected mice. And with the green line, you can see that we have about 5 to 10-fold improvement of the function. We're still far from a normal function, but this result is really giving us some hope that it is possible to intervene during infection and improve hematopoietic stem cell function. So this is, of course, a line of research that we are pursuing very actively at the moment. And for example, Christiana, George, and Marine are using multiple approaches to look at the consequences of strong perturbations of the inflammatory milieu within the bone marrow. And a new thing that we have started to do is to start gaining even wider images of bone marrow tissue, and we can now look at entire bones that we clarify and we stain with multiple antibodies. We then acquire these images that, in this case, of a whole femur of a mouse. It takes about a couple of days to acquire these images, but we can now look at 100,000 of cells, and within these images, we can start looking for the stem cells and see where they are and what are they interacting with. So now, for the last part of today's talk, I'd like to tell you about the other line of research that we've been pursuing at Imperial, which is to understand the interaction between healthy and malignant hematopoiesis. And especially, we want to understand why leukemia, which is blood cancer, which is known to grow primarily within the bone marrow, is able over time to outcompete healthy hematopoiesis. And I will come to the biology in a minute, but I'd like to start with this story because this study was driven by Michael Stumpf and Adam McLean when he was PhD student in his group. And we focused on this study in the very early years of me being at Imperial, when we were still setting up all the biological models, which were complex and took a number of years to get going. And one of the things I learned from this study is that there is really a lot of merit into not just worrying about the biology taking a long time to develop, but we can actually use other approaches, and theoretical modeling can actually give us some very interesting findings. So what Adam and Michael were doing was to use a simple mathematical model based on a couple of equations in which they could simulate the competition between healthy and malignant hematopoiesis. So they could generate these simulations in which the equations had parameters that were given specific values, and eventually they would reach an equilibrium in which we had leukemia cells and leukemia stem cells being very abundant within the bone marrow, and hematopoietic stem cells, progenitor cells, and peripheral blood cells eventually being lost. Now, the beauty of the theoretical approach is that not only we could model what happens in the lab, which is the leukemia wins and overtakes the bone marrow, but we could also model the opposite, and we could use the exact same equations and simply change the value of the parameters that were in those equations, and we could model the opposite, and we could have a situation in which after, let's say, some struggle, eventually we would reach an equilibrium where leukemia would be lost and healthy hematopoiesis would be restored. So then we could ask what parameters were important and what parameters needed to have high values in order to achieve this result. And what became obvious, and at the time it was 2013, and we were really only just very much starting to understand what leukemia does to the healthy hematopoietic system. What we found out was that the only critical parameter was that hematopoietic stem cells needed to have a very high self-renewal. And at the time, we didn't entirely know how to interpret this. But then the biological models came along, and we started collecting some results, and so did the field, and these all started to make sense. So I will show you now how this prediction turned out to be truth. So, looking at malignant hematopoiesis, one of the first models we worked with was brought in by Edwin Hawkins, who came for a couple of years in the lab for his last postdoc before moving back to Melbourne, where he's running a very successful lab at the Weihai Institute. And while he was in London, he also had help we had a great collaboration with Dominique Bonnet at the Crick Institute, and this started actually our deep and strong interaction with the Crick Institute and with Louise Purton in Melbourne. And Dominique and Louise helped us validate the findings that we were obtaining from our urine studies into samples from human patients, which of course was very important. So what Edwin was able to do with this mad guitar that he used to keep in my office, which made my office a very interesting place, was to use it to keep himself awake when he would acquire very long time lapses of T-cell acute lymphoblastic leukemia cells, these ones in red here in this movie, so that he could follow them overnight and really discover what these cells were up to within the bone marrow space as the disease was growing. And the unexpected discovery that he made was that these cells were migratory. And so in this movie here, which was taken over the course of about 14 hours, you can see a single leukemia cell that moves about, then it stops for a moment to divide, which is happening now, and we now have two TALL cells, again, moving around the bone marrow space and growing, and within a few days' time, we would have many more of them, and they would lead to the progression of disease. We were not expecting that these cells would be migratory. The other things I didn't expect, having seen that these cells are migratory, was that even though they were migratory, we would still end up having patches of disease. So we would always have some areas in the bone marrow being more heavily infiltrated with disease than others. And actually at the time, talking to a number of clinicians, it turned out that in fact, the situation is the same for human bone marrow. And when multiple biopsies are taken from patients, some may have a higher degree of infiltrations than others. And this is why it's important to monitor a relatively large area of bone marrow space to really understand how the disease is growing. Now, not only the disease would grow a little bit over time, but if we waited a little bit longer, we could now see mice like this one shown in the image on the left, where the leukemia cells had taken over the entirety of the bone marrow space, and the entirety of the bone marrow cavity is now red, full of red leukemia cells. We could then follow the same mouse as it was treated with chemotherapy, and the image on the right, it's incredibly interesting for us because it shows how well chemotherapy works. Here we are after 3 doses of dexamethasone given once daily, and you can see that the vast majority of red cells have disappeared. But the other thing that we learned is that if you look carefully, a number of red cells still remain, and these are chemo-resistant cells. So what this model allowed us to do was to start studying what is the behavior of chemo-resistant cells. And this is where we had another big surprise, which was that these cells are highly migratory. You can see them being resistant to dexamethasone on the left, but they were also resistant to combined therapy, which is more similar to what patients receive in the clinic. So on the right here, the mouse was treated with dexamethasone, vincristine, and asparaginase. And one thing we learned is that the harsher the treatment was, so the more chemotherapy was combined, the more migratory the cells were. And this was the case for human TALL cells as well. Another thing we learned was that if we didn't treat the mice, and we focused on the osteoblast cells, which are again in green here in the movie, we could see that we would reach a point where the cells would start shrinking and blebbing and becoming fewer and fewer. And in fact, within about 48 hours of when these type of movies would be acquired, we would now have a situation in which one of our mice that normally would have green osteoblasts would have lost them entirely. Not only this was the case for mice, but it was the case also for human. Normally in healthy bone marrow, we can stain for osteoblasts at the interface with the bone along the endosteal surface. But in samples from TALL patients, it was pretty much impossible to find such osteoblasts. So this work now was fitting really well with the findings from Michael and Adam's work, which was that perhaps indeed leukemia does affect the ability of HSCs to self-renew because it affects the very cells that we know are important to support their function. So of course we wanted to learn more about this, and to do so, it became very useful to switch our experimental model, and we started studying acute myeloid leukemia, which is a leukemia that is particularly known to cause cytopenias to patients and to affect hematopoietic stem cells. This was work that was driven by Delfín Duarte, a very talented MD who took a break during his medical training to do his PhD here in London, and now is back in Portugal. He has completed his medical training, and again, he's running a very successful lab there. And he had help again from Louise Purton, but also from Ralph Adams, who is expert in bone marrow vasculature, and Gunnar Pressner from the Department of Mathematics, who helped us better understand the interaction between the leukemia cells and the microenvironment. I'm going to make his story very short, but essentially Delfín focused on bone marrow vasculature because we knew that not only osteoblasts were important to support hematopoietic stem cells, but also endothelial cells. And one of the things that he saw was that within the space, the bone marrow space, which was crisscrossed by vessels and endothelial cells, he could see the leukemia cells also being migratory, both prior to giving chemotherapy and after chemotherapy. And an interesting thing that we saw was that if we treated the mice with an inhibitor of the receptor CXCR4, which is called AMD 3100, which was sufficient to stop the migration of TALL cells and even to induce their death, actually not much happened to the acute myeloid leukemia cells. So this is how we started to learn that the type of interaction of the acute myeloid leukemia cells with the bone marrow microenvironment was much more complex than that of TALL cells. One of the things that Delfín saw was that in particular, the AML was leading to loss of vessels that run near endosteal surfaces. So you can see these vessels in yellow in this image on the top left here, running along with the endosteum in gray in a healthy mouse. And you can see these vessels are lost in a mouse that is heavily infiltrated with AML. This is particularly obvious if we look in the metaphyses of the bones, which are very highly vascularized, and they're much less vascularized if there is AML. And we were able to show that if we look in the 20 microns nearest an endosteal surface in human samples, again, in endothelial cells, which are in brown in these images, were lost in these areas. Not only we were losing endosteal vessels, but we were also losing hematopoietic stem cells, and this was again particularly obvious in the metaphyses areas. It wasn't all doom and gloom, because if we treated mice with the ferroxamine, which is an agent, which is an iron chelator, but it is also known to potentiate endosteal vessels, we could have mice that had leukemia, but they also still had many more vessels near the endosteum, and these mice were able to harbor a much higher number of hematopoietic stem cells. So what this work told us was that, again, not only the case of infection, but also in the case of leukemia, we can intervene on the bone marrow microenvironment and help hematopoietic stem cells. But one thing I mentioned at the beginning of this talk is that I would tell you about the competition between healthy and malignant hematopoiesis. And for now, I have only talked about hematopoietic stem cells. We have looked also more widely across progenitor cells, for example. And here, with work combined from multiple people in the group, and again with Ken Duffy at Maynooth University and Tom Weber when he was postdoc with him, we were able to generate mathematical models that allowed us to look more deeply at how healthy and malignant hematopoiesis compete for space within the bone marrow. What we saw was that hematopoietic stem cells, especially the most primitive ones, are the most resistant to leukemia, and they survive the longest. But if we look at a number of early progenitors, and I'm not going to spend time explaining the details of what each population is, what became obvious is that the loss of these cells actually fitted really well with a very simple model of neutral ousting. And we could measure the size of leukemia cells and of healthy cells to the point that we were able to say that actually per each new acute myeloid leukemia cell produced in the mouse, about 1.7 healthy cells had to be lost. And the model fitted the data beautifully. The reason why I'm telling you about this is because after this work, Chiara, a PhD student in the lab, spent quite a bit of time, again, using intravital microscopy, looking at healthy and malignant cells within the bone marrow space. And she generates movies such as the one that I'm about to play here, in which we have healthy hematopoietic cells in green, surrounded by AML cells, which are in this sort of dim, healthy hematopoietic cells are in red, and they're surrounded by acute myeloid leukemia in this dim green. And we could see them being pushed in this space here, which is a vessel within the bone marrow. They would be pushed en masse into this vessel. And together with these kind of movies, Chiara was also able to detect that only in mice that were carrying acute myeloid leukemia cells, we were able to see clusters of healthy cells in circulation. This is something that doesn't happen in the healthy situation. She could also see that this was coupled with vessels in the bone marrow being very leaky. You can see when the vessels are carrying this pink dye, very quickly it extravasates, and we can find it in the parenchyma. So here again, we put some thinking, trying to understand what was happening and what could be the mechanism behind the images and the movies that we were acquiring. And we started thinking that extracellular matrix probably plays a very important role here. Extracellular matrix provides an anchorage for every kind of cell in the body, and also allows blood vessels to not be leaky. So thinking about what regulates extracellular matrix, we decided to treat the mice that had AML with an inhibitor of enzymes that regulate extracellular matrix and degrade it. And these are matrix metalloproteinases. And again, making a long story short, when we treated mice that had AML with prinomastat, which is an inhibitor of matrix metalloproteinases, we had more dead leukemia cells, but most importantly, we always had more hematopoietic stem and progenitor cells, and mice could survive longer if they were treated with chemotherapy. What this is exciting, why this is exciting for us, is because in collaboration with Brunangelo Falini in Perugia and Andrea Marra, who is doing a PhD in between there and here in London, we've been able to look at the expression of matrix metalloproteinases in AML patients, and we can see that a number of them, and this is just an example for MMP2s, are highly expressed in patients, and perhaps we could identify patients that could benefit from inhibition of matrix metalloproteinases. Lastly, we're actually learning that the interaction between leukemia cells and other types of healthy cells, so non-stem and progenitor cells, is highly complex. And I will show you an example here from work driven by Dina Pospori, looking at the interaction between AML cells in red and T cells, where we are learning that at early stages of disease, we can literally see the battle between leukemia cells and T cells, forming clusters and forming opposing fronts within the bone marrow space. But we also know that as leukemia grows and we start having larger clusters of leukemia cells within the bone marrow space, then the T cells end up being dispersed, and eventually leukemia does outcompete the immune system and healthy cells. And so, of course, we are spending more effort looking at ways to target the bone marrow microenvironment or target leukemia bone marrow microenvironment interactions, so that we can hopefully find clues towards the development of improved leukemia therapies. And I'm pretty much about to stop here. I just thought I would mention where we work, which it has been in the Fleming building in the South Kensington campus of Imperial College, where we will keep working for the foreseeable future. This is a wonderful place where we are next door scientists that work with a number of pathogens, with a number of developmental systems, but also physicists, mathematicians, engineers that can help us have a different look at the systems that we are looking at. We're also working at the Francis Crick Institute, and especially there we're excited now to have started work with Ilaria Malanki, with whom we will start engineering bone marrow microenvironments to see whether we can affect, for example, stem cell function or leukemia growth. And we're also very excited to start working more closely with the Hematology Center at Imperial, based at the Hammersmith campus, where I think we will have tremendous opportunities to have great interactions and exchanges from the bench to the bedside, and the other way around through all the clinicians that work in the hospital there. So I'll just finish with a quick thank you, first of all, to all of you listening me ramble for probably more time than I should have. I need to thank all the funders that have allowed us to perform the work over the years. And I guess I'll just finish off with a special thank to my husband, who's been with me since the PhD times and has really supported me throughout all these years, and with whom just a couple of years ago I started the biggest adventure yet, which is to help this little human grow, and we'll see where she will end up.
So thank you very much, Cristina, for an absolutely outstanding lecture. There's comments on the chat line like sort of incredible images, etc. But I won't say any more at this point because we go straight into the Q&A session. And to just give you a chance to pause, I will actually just comment that there is a comment on the chat line which has, say, number of likes saying not a question, more of a heartfelt thank you to you and your team for all of your research into beating leukemia. So what I am planning to do, Cristina, and for those who are attending, is I have selected a few of the questions that came in advance, and I'm targeting first the young people in the audience. So the first question is from Medusa, who is a school student, and she says, do stem cell banks have enough donors, especially during the COVID-19 crisis?
So, because I'm not a clinician myself, so I'm not in the hospital in the first place, I'm in a bit of a difficult situation to comment about the impact of COVID on banks. But whether, for the general question, whether we have enough samples, the answer is complex. It is very important that we have as many donors as possible, especially across communities and across different ethnicities. We know that it is harder, for example, to find matched bone marrow donors for minorities. And so it is important to have more and more donors and more samples. At the same time, there is also a very active line of research that is working towards expanding hematopoietic stem cells. And while I say that we cannot do it very effectively yet, it doesn't mean that we will not be able to do it in future. And in fact, we think that some of our work looking at hematopoietic stem cells in the bone marrow is in fact teaching us what is important that we do to make stem cells grow in vitro so that we will be able to treat more patients with the samples that we have available. For example, seeing the stem cells wriggle and have these complex and very dynamic interactions with the surrounding is making us think that simply putting them in plastic is not enough. We need to provide them with much more complex types of stimulation. And there are also even efforts that are put into generating banks of somatic cells harvested from donors, and then these cells can be turned into pluripotent stem cells, and these cells can then be turned into cells that are needed for regenerative medicine, such as, for example, hematopoietic stem cells. Now, there are several steps along the way here, and there are several challenges. But for example, I know Shinya Yamanaka for a number of years in Japan has been driving a very large effort into generating a biobank of induced pluripotent stem cells that would match the most common and as many as possible of the HLA haplotypes that are present in the population. So things will keep improving.
Okay, thank you, Cristina. So we'll move now to a PhD student, Alexander, who is a mathematician by training, and he's asking what special mathematical skills are needed in order to help your kind of research.
I would say probably not necessarily the most special mathematical skills, but some skills that are very important for the type of interdisciplinary work that I presented here are, for sure, patience, the ability to explain what seems obvious to someone that thinks is not obvious at all, and then also the ability to trust the other person. it was incredibly interesting for me working with Charles Lim. We spent years, to be honest, with me going to him saying, why is your microscope not showing this or not showing that? Why cannot we add, a couple other cell types or a couple other channels without having any idea of how challenging it would be to do that? And he spent a few years asking me, why do you have to use all these markers to identify hematopoietic stem cells? But eventually we figured that each of us knew what we were doing, and so we could work together, and we actually got to where we were. And I'm still in touch with Charles. In fact, actually, I think he has logged in today, so if you're there, hi Charles, and thank you. And it's still absolutely wonderful to discuss science with him. So being very open-minded and very, very, very flexible and prepared to explain the obvious is absolutely important. And then I think modeling has been incredibly useful to us. And I know that that can be done in many ways. We are starting to use artificial intelligence now. So this is a field that is definitely starting to meet biology really well. And one other field of math that we're starting to interact with is data spatial statistics. And this is because we're able to see more and more cell types within the bone marrow. We want to understand whether their localization and interactions matters. And so it's very important that we have some very state-of-the-art and complex statistical methods that allow us to test these data.
Okay, so next question, Cristina, is from your postdoc mentor. How do age and sex affect the parameters you evaluated?
Thank you, David. Very good question. They do. They certainly do. To a certain extent, we know they do. To a certain extent, there is still a huge lot that we need to learn. We know that in mice, for example, hematopoiesis changes as the mice become adult, and I'm sure the case is for humans as well. So at around 3 weeks of age in the mice, hematopoietic stem cells become a lot more quiescent, for example. And then as the mice age, again, things change. And we know that in older mice and in older humans, hematopoietic stem cells are more proliferative and they're less functional. And we know that the microenvironment in the bone marrow also changes. For example, the bone marrow is not as well vascularized as in younger subjects. And Ralph Adams, for example, has shown with Angelico Zumber that if endosteal vessels are made more abundant and better functional, then hematopoietic stem cells can be rescued. So definitely a lot of changes happen with age. We also know that hematopoiesis becomes more and more clonal. So while we are born with a certain number of hematopoietic stem cells, the pool of hematopoietic stem cells becomes more homogeneous, and when we lose clones of stem cells over time. And these can have implications for the development of hematological malignancies. It's very important that we have to understand that. As for sex, there are some quirks. So for example, we know that female mice respond to parathyroid hormone treatment really well, but male mice not so well, and we don't quite know why. So I will just leave it at that, saying that there is still a huge lot that we don't understand about the effect of sex on hematopoietic stem cells, and future will tell us. We're starting to be able to compare.
Sorry, Christine, I was going to say thank you for that. I'm just going to ask 2 more quick questions, and I have to apologise that there are a lot of questions on the chat line. Remarkably, there are well over 200 people attending this lecture, which is amazing. So I do hope that many of you who put questions on the chat line early will have found the questions at least in part answered as we progressed through. What I hope will be a quick question, Cristina, how do you know which cells are stem cells? That question is from Philip, and there are a lot of likes for it. So how do you know which are stem cells?
So for the hematopoietic stem cells, we are lucky that we can use flow cytometry, which allows us to see proteins that are on the surface of the cells. And we can identify stem cells because they express on their surface a whole lot of proteins, but not a whole lot of others. And so by using combinations of antibodies, we can recognize which cells are stem cells. Of course, this means that we can do it once the cells are out of the bone marrow, and we are looking at them in the tube. But from that, we can then look at what other genes they express. And over the last number of years, actually, for example, a number of transgenic reporter mice have been generated, so that now we have stem and progenitor cell populations that more or less correspond to the populations we identify with flow cytometry that express fluorescent reporters. And we can now look at the bone marrow of these mice, and we can see, for example, what happens to these cells during infection or as leukemia progresses, and we can literally follow them and see what they are doing. And maybe in the next public lecture, there will be something about that.
Okay, so the final question is a teaching-related question, again with several likes. When you teach stem cells, regeneration and aging, is it more human-based or animal-related?
I think it is very important that when I teach, I transmit the excitement for the discoveries that have been made. And because my work is primarily based on murine studies, I guess my lectures contain a lot more murine biology than human biology. Having said that, the hematopoietic system of mouse and human is quite similar. A number of the discoveries we have made have been validated in humans, and a number of the things that we find from mice remain valid for humans as well. But of course, we go well beyond what we do within the lab, and so we look at a number of human studies as well. And especially actually with current technology, it is possible to learn from humans things that were absolutely unthinkable until a few years ago. And people can now, sequence human stem and progenitor cells and identify the families of cells, so which lymphocytes or myeloid cells are connected because they derive from the same hematopoietic stem cells, and how many hematopoietic stem cells there are in a certain individual, and whether the individual is developing clonal hematopoiesis or not. So it's now becoming possible to understand how hematopoiesis is happening in real time in people as well. So I think that probably over the next few years, there will be more and more human coming into my lectures.
Okay, thank you very much, Cristina. And I'd like to hand over now to Miriam Haltali, who is very kindly giving the vote of thanks. I just want to say that Miriam, you will have seen on Cristina's slide, she did her PhD with Cristina, finishing in 2018, is now a postdoctoral fellow at the Cambridge Stem Cell Institute. So over to you, Miriam.
Thank you, Anne. And thank you for the opportunity to help celebrate Cristina today. As mentioned, I did my PhD and my first postdoc under Cristina's supervision. And I think that through this lecture, you can understand what appealed to me and many others when considering joining this group. As you've seen, Cristina has made truly paradigm-shifting contributions to understanding the dynamic cellular processes that regulate hematopoietic and malignant cells in the bone marrow through this pioneering use of intravital microscopy. And this has not only generated the stunning images that we've witnessed here today, but has also been really key for extracting important and novel information that has informed some new hypotheses in the field. She's shown this rare ability to successfully tackle widely recognized technical challenges to advance the field of stem cell biology using a really interdisciplinary approach. And this is something that really makes Cristina stand out from the pack. So I was a new master's student when I first met Cristina, and I immediately noticed her infectious enthusiasm and excitement for the research that was happening in her lab. A year later, when I came to choosing where to carry out my PhD, I knew that her positive, collaborative, and supportive approach would see me through this new challenge. Now, working in research can be really hard, even at the best of times, and often there are so many lows before you experience any of these remarkable highs that Cristina has presented here today. And from speaking to some of my colleagues and people that have worked with Chris previously, one of the most common opinions is that she has this amazing ability to empathize in these moments, in these low moments. And she will often share a story or two from her own PhD or postdoc days, where she will claim that she had it worse than we did, and she will definitely find a way to make us feel that we're not alone in this and it will be okay. She always made us look look at the bright side, especially when all we can see is darkness. And many would agree that she is unafraid to take projects in completely new directions when things have not gone completely according to plan. She's a fantastic listener, and her door has always been open for discussion. Cristina puts really great importance in maintaining a productive working environment and good team spirit. And this is demonstrated by the range of group activities that she plans, from lab retreats in the countryside to pasta making in her kitchen. She's incredibly open to new thoughts and ideas, and she truly values each opinion that is shared in the lab, whether it comes from an undergraduate, summer student, or a senior postdoc. Everyone is made to feel that they're an important part of the team and that we're all actively contributing to writing this incredible group story. Now, beyond the amazing science, awards, public outreach contributions, teaching, and the list goes on, Cristina still takes out the time to thoroughly mentor her junior researchers and push them to reach their full potential. She always has plenty of exciting opportunities up her sleeve for anyone who's willing to put in some extra work and gain a bit more experience. And she'll always be there to support you from start to finish. And this support goes not only for those who work directly under her supervision, but Cristina is arguably one of the most approachable, friendly, and helpful senior researchers in our field. And there are plenty of people who could vouch for this and attribute many aspects of their own success to her selfless mentoring and guidance. From this, there's really lots more that can be said, but this would take way more than the 5 minutes that I've been given to speak today. But it's safe to say that during her time here at the college, Cristina has composed the perfect niche to support the long-term production of some truly groundbreaking science. So I'll come back to the task at hand. I need to thank Professor Cristina Lo Celso for this inspiring inaugural lecture. Thank you for your continued guidance and mentorship. We're all looking forward to seeing where you take things next as you step into this new role. And we really hope that we'll be able to celebrate with you in person one day in the not too distant future. Thanks also to Professor Andal for chairing the lecture and introducing Cristina today, and to our events team, Sam Phillips, Christine Taylor, and Amy Brown, who've put on this great event despite the challenging circumstances this year. And a big thank you to you, everyone in the audience who's joined us and asked loads of really interesting questions. The last thing I need to mention is that the next lecture in this series will be taking place on the 27th of January, where we'll have Professor Bjorn Schuller, who will be charting the history of human-centered AI in his inaugural lecture, titled AI for You and I. So that's all from us. Thank you very much for joining, everybody.